And without any further delay, let's jump on to our first speaker, who is Dr. Hua Pu. Hua is a postdoctoral researcher in Target Discovery Institute, University of Oxford. He works on the transcriptional factors of circadian clock to discover drug targets and develop therapeutic molecules. Hua received his PhD in developmental biology at the Institute of Botany, Chinese Academy of Sciences, and subsequently he shifted to University of Oxford to study this. He will be talking to us about BMAL1 targeting for development of small molecules to control chronic inflammation. Hua.
Thank you for the introduction. Hello, everyone. I would like to share a story about drug BMAL1. Your body has an internal 24-hour clock that controls sleep-wake cycles and various biological processes. This is a well-known circadian clock. Circadian clock at molecular level is interlocking transcription, translation, feedback loops. In mammals, the transcription factor BMAL1, brain and muscle unlike one, heterodimerizes with its partner to locate at the central node of these loops. BMAL1 clock drives the expression of cry and purr. Cry, purr, and then bind to BMAL clock to stop its own expression. Following the degradation of cry1, the free BMAL clock then drives another round of expression. So this cycle roughly takes 24 hours, which forms a cell-autonomous circadian clock. BMAL1 contains 4 domains in the structure: one DNA binding domain at the N-terminal, one transcription activation domain at the C-terminal, and 2 path domains in the middle. The term of path domain was named after 3 proteins, PER and SYN, in which it was first discovered. We now know BMAL1 is associated with many diseases, including chronic inflammation, chronic kidney disease, cardiovascular disease, and cancer. However, intervening of these diseases through BMAL1 is still not possible. Our story about BMAL1 starts from high-throughput screening. We obtained the first known hit for BMAL1 in summer-shift assay. We confirmed the direct binding of this hit with BMAL1 by SPR, which gives a big number about 172 macromolar. We then performed hit lead optimization and developed a lead compound, CCM, core circadian modulator. CCM bound in the central cavity of the path B domain of BMAL1. You can see here, the cartoon represents the BMAL1 protein, and the stick represents the CCM compound. Surprisingly, CCM expands the volume of the cavity for about 3 times. This induces a lot of conformational changes, including pushing out a rigid helix, FF, which is crucial for the cellular function. Mutation of glutamine 379 here in the figure to alanine was reported to reduce BMAL1 binding to nucleosome. Our finding confirmed this. We took an unbiased approach, employing random experiment to identify all interactions of the endogenous BMAL1. As you can see here in the figure, the interaction of BMO1 with other known importance like clock, MPAS2, PERS, and CRIS was unaffected, while the interaction of BMO1 with histone H2A was significantly changed. The impaired association between BMO1 and histone result in the repression of circadian clock at cellular level. CCM mainly downregulates the expression of core circadian genes, including CRA1 and PER2. CCM also downregulates phagocytic pathway in macrophage. The phagocytosis genes were mainly downregulated in macrophage on the CCM treatment. The beauty of CCM compound is that it represses the amplitude of circadian clock, but keeps the level of BMO1 itself unaffected. As you can see here in the figure, the level of BMO1 itself remains unaffected on the CCM treatment at both iron level and protein level. This is not the case for all other clock modulators, including small interfering RNA and compound targeting other circadian proteins. In addition to offering a unique tool for circadian research, CCM provides the first clinical demonstration that therapeutic opportunities through BMO1 modulation could become possible. We explored the therapeutic potential of CCM compound by using unsequencing in uterus cells. Gene ontology generated in our representation analysis highlight cartilage diseases. One of the cartilage disease, osteoarthritis, draws our attention. Osteoarthritis is a highly age-related disease with degeneration of cartilage in pathological tissues. Globally, 595 million people had osteoarthritis in 2020, and this number was predicted to reach 1 billion by 2015. There has no cure for osteoarthritis and limited option for symptom medication. Patients with osteoarthritis have symptoms following circadian rhythm with time-day varies in pain and stiffness. BMO1 was reported to control the cartilage homeostasis. Knockout BMO1 causes osteoarthritis in mice with degeneration of near articular cartilage. In our experiment, one of the genes altered by CCM through BMO1 is MGP. MGP is required for cartilage maintenance. Impaired expression of MGP gene is associated with increased risk for osteoarthritis. On the other hand, CCM also downregulates the inflammatory cytokines, including interleukin-6 and TNF-alpha. Targeting BMO1 could benefit osteoarthritis by upregulating cartilage maintenance and downregulating inflammatory pathway. What's more, we obtained further opportunities for drugging BMO1. We got a second series of BMO1 compound. These compounds bind to the to the central cavity, but it pushes the tyrosine 314 in the pocket, which induces a lot of conformational changes in beta strand A, B, and H. This is a total different conformation change compared to what we see for CCM. On the other hand, we also got hit for the path A domain of BMO1, which is far away from the binding site of CCM. To summarize, we obtained a hit for BMO1 and then developed an inverse agonist, CCM, called circadian modulator. Use CCM in your study if you would like to repress the circadian clock, but at the same time keep stable the background interaction among circadian proteins. Free CCM compound is available upon request. What's more, we think there is a compelling case that BMO1 modulation by CCM alters the expression of crucial genes in osteoarthritis. This marks the therapeutic potential of BMO1 compounds. Last but not least, we obtained further opportunities for drugging BMO1. These include a different chemotype for BMO1 plus B and new hit for BMO1 plus A. This is all for my presentation. Here is a figure for my group. I would like to thank Professor Frederick Stiniged, my colleagues, and our collaborators. This project received research funding from Wellcome Trust, NIH, and a travel grant from Biochemical Society. Thank you.
Thank you very much, Hua. We might have time for one question, please, if anyone has a question. Oh, Mike, here.
That was a lovely talk. I've led a group on melatonin for chronobiology, and I can assure you it's a really much more important aspect than you think. Are you considering looking to see whether melatonin agonists can synergize with BML, with your compound in vivo?
Well, actually, it's different thing, actually. Melatonin is a central clock. Well, BMO1 exists in the central and the periphery. Well, here we actually mainly focus on the periphery clock.
But both can drive both, right? So it's a synergy which is really quite important.
Yeah, it's the intertalking, but the central to the periphery definitely is important. But melatonin is well studied, and they already have a lot clinical trial and also approved like treatment.
Yeah, like Ameliorates, which Servier brought out. So it really is a powerful mechanism for immunology as well.
Thank you very much, Hua.
All right, so our second speaker for this session is Kieran Feltham, who's going to talk about phenotypic screening of AstraZeneca's annotated compound library for the prevention of hypertrophic scars.
Hello, everyone. Firstly, thank you to the organisers for allowing me to share my project with you guys today. Today I'll be talking about a joint project in collaboration with AstraZeneca using one of their annotated compound libraries for the prevention of hypertrophic scars. So roughly 11 million burn injuries occur every year worldwide, and up to 91% of partial thickness and full thickness burn wounds will result in hypertrophic scarring. This is not only cosmetic, but also functional consequences. This includes neuropathic pain and also contractures, which you can see at the bottom, can really inhibit movement of the patients. Scarring also results from fibrosis, where conventional wound healing becomes dysregulated. So there's currently no medication available for the prevention of hypertrophic scar formation. The market size currently is estimated to be just over $32 billion, and this is on target to more than double within the next decade. Also, the current pipeline for new anti-scar medications is weak and suffers from high attrition rates in phase 2 clinical trials. This is roughly 80% across all fibrotic diseases. A brief overview of the pathophysiology of fibrosis. Tissue injury occurs, on the epithelial cell layer. This leads to the activation of platelets and the recruitment and activation of inflammatory cells. These in turn release cytokines, growth factors, interleukins, and such. and these cytokines aid in the activation of fibroblasts into myofibroblasts, which is our disease phenotype. They produce excess collagen and other matrix proteins, which are then deposited into the extracellular matrix. This aids and drives scarring. So our aim was to identify compounds and/or targets that prevent transformation of human primary dermal fibroblasts to myofibroblasts. We use transforming growth factor beta-1 as our cytokine to induce this change. It's well documented, and it's a very potent cytokine for this. So we initially obtained burned scar tissue from St Andrews Centre for Plastic Burns and Surgery, and Scarring, and then isolated primary human fibroblasts from the tissue. We then used our phenotypic screening assay to differentiate these into myofibroblasts using TGF-beta1, and also sent off fibroblasts and myofibroblasts to RNA-seq analysis just to confirm that our targets were being expressed by our cells. We then optimized and validated said assay before screening roughly 3,500 annotated compounds from AstraZeneca's library, with the intention of completing secondary assays to confirm the antifibrotic effect of candidate compounds. So firstly, our development of our phenotypic screening assay was validated using 10 nanograms per mil of TGF-beta1. As you can see, there was minimal variation and a good fold change between our cells, which allowed us to achieve a Z factor of 0.63. AstraZeneca's annotated small molecule library consisted of 4,000 annotated tool compounds with an affinity of less than 100 nanomolar against roughly 1,900 human targets. These consisted of preclinical and clinical candidates, compounds profiled extensively in selectivity panels, tool compounds and marketed drugs, and to the right you can see the classifications. So yeah, initially we screened 3,500 compounds at 0.1, 1 and 10 micromolar. This was in singlet. 644 of these compounds produced a concentration response curve for which we could get an IC50 from. They were then further excluded if specific inhibition and viability thresholds are not met. This was namely more than 80% viability at 10 micromolar, as well as more than 80% inhibition at 10 micromolar. 198 compounds were taken forward and then further triaged based on target expression from the RNA-seq data, potency from our concentration response curves, and selectivity data, which was provided by AstraZeneca. 65 compounds were then taken forward, and full 8 to 11 point CRCs were then made in triplicate in 3 different cell lines. AstraZeneca also provided us with 159 structurally similar near-neighbor compounds. Basically, we wanted to see if structurally similar near-neighbor compounds could also provide a strong antifibrotic effect compared to their primary compound. These were also done in full 8-point CRCs in triplicate in 3 different cell lines. And from this, we identified 12 targets that were repeatedly acted upon. So these are some examples of the concentration response curves from the 65 hits. So these 4 compounds were some of our most potent, with 3 of the 4 maintaining high viability throughout, and all their IC50s were within the nanomolar range. And then next, some examples of hit compounds. So this is compound E and their near neighbors. Now, compound E was the only compound to have near neighbors that essentially gave us concentration response curves and suitable IC50s. I think the best one for us was compound E3. And then out of the 159, it was much more like this, where there was a severe loss of potency from the near neighbor compounds, and quite a few of them were actually showing pro-fibrotic effects, not anti-fibrotic. So in conclusion, we screened roughly 3,500 compounds from AstraZeneca's annotated compound library, and then identified 65 hit compounds based on target expression, potency, and selectivity. Inhibition in a concentration-dependent manner was achieved in only 1, at the level we were interested in, Of the 159 near neighbours selected for testing, suggesting there is a limited structure-activity relationship, kind of highlighting the complexity of fibrosis. And we are currently validating the 12 targets identified using secondary assays. So this is collagen contraction and ECM production assays. Finally, my acknowledgements. So firstly, the fibrosis team at Anglia Ruskin University, as well as the team at AstraZeneca and St Andrews Centre for Plastic Surgery and Burns. Thank you.
Thank you. We have time for questions for Kieran. Maybe I can start. So, do you have any thoughts for why the near neighbours were so poor?
Yeah, so we was hoping it would be a little bit better than what occurred. A lot of the compounds from the hit compounds are acting on multiple targets. So we think that even though there's slight structural differences, because it's such a highly conserved process, wound healing, obviously they have to be hitting certain targets to have that antifibrotic effect. If there's one or two pathways that aren't being hit at certain concentrations, it could just be the reason why, yeah.
So instead of near neighbours, I mean, it's an annotated library, so did you look at other known compounds with activity at the same target?
So not yet. What we did do as well with some of the RNA-seq data is, so we had the initial, there must be at least 80% viability, but as you saw, one compound went below that. So we actually took some compounds in from the RNA-seq data, some of the compounds that were very, very potent against some of the targets that were highly expressed. But yeah, we haven't gone any further with that.
Okay, I think we have a question on the front here.
Thank you. Thank you for the great presentation. But I think you mentioned that there is no medication for hypertrophic scar tissue.
So,
but there is a corticosteroid, triamcinolone, but they inject it in the tissue.
Yeah.
Also, it's not a medication mark. There is also laser therapy, radiotherapy.
Yep.
So I just want to ask, what do you think about the corticosteroids for this use? And the second question is, you observed TGF-beta levels.
Yeah.
And what about other stockings like interleukin-1 beta, interleukin-6, interleukin-10? Thank you.
Okay, so first of all, yeah, so there are definitely medications and ways to treat fibrosis. It was more, there's not something out there that will fully inhibit the scarring completely. Apologies, do you mind repeating that first question again for me?
My first question is, there are corticosteroids for this hypertrophic scar tissue. But you mentioned that there is no medication for this.
So there is medication, but it's more just to fully halt the pathology of fibrosis itself. Steroids are well used in fibrosis, especially topical. I think as a group, it's something we're not necessarily interested in too much, because with wounds as well and steroids, you don't want to use them for prolonged periods of time with steroids. Big burn wounds, for example, it's going to take a while to heal. They are very effective, but yeah.
And what about other cytokines?
Yes, so we have tried others. So we tried, as part of the validation, so we used TGF-beta-1, also CTF and PDGF. TGF is just very, very well documented and used. It's also very potent. When validating the assay, we got a much better Z factor using TGF compared to the others. So for the assay, we thought it was the best cytokine to use.
We have time for one more question in the front.
Thanks for your talk, very nice. Could you give us a bit of perspective on why you used fibroblasts from scar tissue instead of using commercially available ones? Yeah, that'd be interesting.
Yeah, so we're fortunate enough to have access to primary burn wound tissue. What we find, or I guess in general as well, is you always want to get as close as possible to the real thing, really. Especially burn scar tissue, and the fibroblasts, when we use that from like primary tissue that maybe isn't burn scar tissue, it is affected differently because it's just, yeah, it's like the wound, the trauma, the way the cells respond. So we just want to try and be as physiologically accurate as possible to when it comes to testing these compounds.
Yeah, I mean, really good rationale, but I'm very curious, though, as to whether, you know, a commercial fibroblast would respond to your hit there.
I'm not sure. As a group, we just used the primary cells, but it is, yeah, it is a good point.
All right, join me in thanking Kieran.
Our next speaker is Dr. Joohyun Nam. Dr. Nam is a professor at Dongguk University College of Medicine's Department of Physiology and the vice director of its Channelopathy Research Center. And Dr. Nam would be talking to us today about Asarinin as a potent natural TRPV3 inhibitor.
Thank you for your kind introduction. This is the first time to participate British Pharmacology meeting, so I'm deeply honored to give this opportunity to present my work in front of the British Pharmacological Society members. Today, I would like to talk about my recent work regarding the in silico screening of a natural TRPV3 inhibitor for skin inflammatory disease. I would like to start with a brief introduction of the TRPV3 physiology and pathophysiology. TRPV3 is a calcium-permeable, non-selective cation channel which can activate by warm temperature above the 33 degrees and various chemical agonists such as 2-APB and natural compounds, carvacrol. TRPV3 is abundantly expressed in the keratinocyte, which involves in epidermal differentiation, a process crucial for skin barrier formation and also hair growth. However, the activation of the TRPV3 can release the pro-inflammatory cytokine in keratinocyte. Therefore, abnormal TRPV3 hyperactivation or gain-of-function mutation can cause the severe inflammatory skin disease such as the Olmsted syndrome, which is the rare genetic disease by gain-of-function TRPV3 mutations in humans, atopic dermatitis, and psoriasis. Therefore, our research goal is to identify novel TRPV3 inhibitors via in silico screening for inflammatory skin disorders. This slide summarized our in-house in silico screening workflow. By collaborating with the computational biochemist, we started our screening with non-also binding site on TRPV3. First, in step 1, we performed the molecular docking simulation using Aurorovina on a library of the 1,063 natural compounds. As a result, we found 74 candidates, and in step 2, we calculated the residue interaction similarity score using protein ligand interaction profiler to see whether this candidate can share similar binding profile with the also. So we can refine the 20 candidates. And then in step 3, we performed the manual inspection of these compounds for chemically plausibility and see whether it meets the Lipinski rule of 5. So ultimately, we yielded the 4 final hit compounds. And next, we performed the experimental validation of these compounds. So we performed whole cell patch clamp recording in TRPV3 overexpressing HEC29 T cells. As you can see, the only Asarinin showed the inhibitory effects on the TRPV3. The Asarinin significantly inhibits the TRPV3 currents induced by various stimuli such as 2-APB, carvacrol, and heat. And we also performed the selectivity test for TRPV subtypes. As you can see that the Asarinin only can inhibit the TRPV3 among the TRPV subtype and TRPV1. As I mentioned earlier, TRPV3 is abundantly expressed in the keratinocyte, so we double-checked the mRNA expression of the TRP channels in normal human primary keratinocytes. So as you can see this bar graph, TRPV3 showed the highest expression profile among the calcium-permeable TRP subtypes, such as the TRPV, TRPA, and TRPC subfamily. So the agonist cocktail of the TRPV3 can induce the huge amounts of the endogenous TRPV3-like currents, and Asarinin significantly inhibits in a dose-dependent manner with the IC50 value of the 12.2 micromole. And Asarinin can also suppress the calcium influx induced by the TRPV3 agonist cocktail, and subsequently reduces the key inflammatory cytokine release in human keratinocytes. And we asked whether Asarinin can also inhibit gain-of-function mutation of the TRPV3. These mutants maintain constant open state, which leads to the continuous calcium influx. So consequently, when we overexpress the HEC cell, the cells spontaneously undergo the apoptosis. So to perform the experiments, we have to reduce the cDNA amounts during our transfection, and also reduce the extracellular calcium concentration in the culture medium. So as you can see, this graph, we had to simply adding the physiological concentration of the calcium evoke the TRPV3 currents and fuse intracellular calcium increase. But the Asarinin can significantly inhibit these mutant currents and intracellular calcium increase. And moreover, Asarinin can prevent the death of the HEC cells expressing the gain-of-function mutants in a dose-dependent manner with the EC50 of the 18 and 11 micromole, which are consistent with the IC50 of the TRPV3 current in HEC cell. And next, to confirm is the therapeutic potential in vivo. We tested the Asarinin in DNCB-induced apoptotic dermatitis-like mouse model. So as you can see, this graph, the topical application of the Asarinin significantly reduced the skin severity symptom score, which comparable to the efficacy of the tacrolimus, a potent calcineurin inhibitor widely used in clinic. And next, we also assessed the epidermal hyperplasia, mast cell infiltration, and mRNA expression of the inflammatory cytokine in skin tissues. The application of the Asarinin significantly reduced the epidermis thickness and the mast cell differentiation. And moreover, in case of the mRNA expression of the key inflammatory cytokines inhibited by the dose-dependent manner, and also it surpassed the effects of the tacrolimus efficacy on mRNA expression level. In summary, we highlighted 3 key findings. So we successfully identified the potential TRPV3 inhibitors from a large natural compounds library using the in silico screening. Second, we validated that Asarinin selectively inhibits the TRPV3 channel activity calcium influx and inflammatory response in human keratinocyte. Third, Asarinin showed the efficacy in ameliorating symptoms in atopic dermatitis-like mouse model. And crucially, as shown on the right, the Asarinin exhibits a much more favorable cytotoxicity profile compared to the other known inhibitors. So more notably, Asarinin showed no significant cytotoxicity even when the concentration was increased to the millimolar range. So therefore, we propose that the Asarinin is a promising candidate for targeting TRPV3-mediated skin disorder, particularly due to its safety and its efficacy. This work is collaborating with Professor Lee's lab from the Catholic University of Korea, and thank you for the attention.
In case anyone has any questions, please come forward. Meanwhile, I can start. I was wondering, Asarinin has 4 chiral centers. If you look at the structures, that kind of makes it synthetically very, very complex as you kind of expand on the SAR. Did you try any other related molecules as a proof of concept to demonstrate the specificity of Asarinin's action?
Actually, I'm an electrophysiologist, so I did not think about these kinds of the matter. So these days, we are keep finding medicinal chemists to make the synthetic compounds of the Asarinin.
Thank you. Please.
Thank you for this beautiful presentation. But I just want to understand the motivation of this study, because you showed us at the first slide, Ostol is a natural coumarin compound. Mm-hmm. Yeah, please. Yeah, Ostol is a natural coumarin, but Asarinin is a lignan type coumarin. So I don't understand how you connect them and began to study with Asarinin. Is there any connection? Yeah, why did you show us Ostol and natural coumarin and that study, but you used the Asarinin as a lignan compound, a compound belonging to a different family?
Yeah, so actually, I'm sorry if I didn't know that, but actually the key concept is that by the cryo-EM studies, the binding site is already revealed for the TRPV3 inhibitors. The advantage of the molecular docking simulation is that it's based on the lock and key model, like the Lego block. So actually, even if it's a different chemical, but if it can fit in the binding site, we can easily predict whether it can bind to the binding site or not.
So you are focusing on, I think, the natural.
Yeah, so I'm focusing on the natural compound which can inhibit the TRPV3.
Even they have not a common structure. Also.
Yeah, yes.
Okay, thank you.
I was wondering, continuing on the question that that gentleman was posing, what is the activity of Asarinin compared to when you compare it with the molecule Ostol? Would you have controls in the?
Actually, by comparing with the IC50 values, the known other natural derived compounds, our Asarinin showed a much more potent, significantly potent than the Ostol, but it's not potent like the alpha mangosteen. But alpha mangosteen shows a significant cytotoxicity, but our Asarinin have no cytotoxicity, so it's much more superb than the other natural compounds.
Wonderful, wonderful. Thank you so much for the wonderful talk. Let's join, please join me in congratulating Joohyun.
Thank you.
All right, so our next speaker is Dr. Elif Alan Rebelhiak. Albrecht, sorry. Speaking about transglutaminase 2, a potential therapeutic target for diabetes-induced sexual dysfunction.
Hello everyone, my name is Elif Alan Albayrak. I am working with Professor of Simonson's research group at Aarhus University, and today I will present my Marie Curie postdoctoral fellowship project on transglutaminase 2 or TG2 as a potential therapeutic target for diabetes-induced sexual dysfunction. Worldwide, more than 500 million people live with diabetes. Approximately half of them experience sexual dysfunction in both sexes. However, if you look at the last 25 years of literature, diabetic male sexual dysfunction has been studied almost 10 times more than female diabetic sexual dysfunction. And this clear imbalance highlights an urgent need in more research for female, which is one of the main motivations for this project. If you look at what is normal sexual function, in female and male, corpus cavernosum in clitoris and penis is composed of smooth muscle and artery. It fills with blood during arousal, and this blood mainly comes from internal pudendal artery. Mechanistically, pathways are very similar. Nitric oxide release from endothelium and nerves relaxes smooth muscle, causes penile and clitoral engorgement, and in women also lubrication. So vascular function is crucial for sexual function. Chronic high blood glucose level in diabetes causes vasculopathy, fibrosis, and neuropathy. In genital tissues, it decreases blood supply, increases tissue stiffness, and impairs nervous system, so leading to sexual dysfunction. In a bit more detail, high blood glucose damages endothelium, decreases nitric oxide production, impairs vasodilatation, and blood flow to the genital tissues. Additionally, it upregulates fibrotic ECM components such as TGF-beta1, CTGF, collagen-1, and fibronectin, causing vaginal and penile fibrosis, which is further amplified by hypoxia. And these responses are also induced by glycation end products, which promote oxidative stress. Additionally, in women, it decreases mucosal hydration and lubrication and causes dyspareunia. In this project, we mainly focused on fibrotic and vascular component of this pathway. Regarding treatment options in men, PDE5 inhibitors such as sildenafil rely on a healthy endothelium. So in diabetes, they are ineffective in more than half of patients. In female, options like estrogen, testosterone, and flibanserin have serious side effects, limited efficacy, and importantly, none of these therapies address the vascular component or fibrotic process of female sexual dysfunction. So there is a clear unmet need for advanced mechanistic studies to identify better therapeutic targets. Here, where TG2 comes in. TG2 is a multifunctional enzyme. It can switch between two conformations. In its close GTP-bound state, it promotes signaling pathways that helps vascular regulation. In its open calcium-bound state, it has transamidase activity, cross-linked structural proteins, and causes fibrosis. TG2 has been implicated in diabetes-induced oxidative stress, endothelial dysfunction, and fibrosis. In this project, we use LDN, reversible allosteric modulator. It promotes its close conformation. And we compared its effect with Zeton, which is an irreversible active site inhibitor that locks the enzyme in its open conformation. So none of its open or closed state activities can take place. Putting this together, our hypothesis is TG2 contributes to diabetes-induced sexual dysfunction, and by promoting its close conformation with LDN, we expect to restore nitric oxide-mediated relaxation and reduce fibrosis, and finally, improve sexual function. To investigate TG2-driven fibrosis in genital tissues, we used primary human uterine fibroblasts and human dermal fibroblasts from neonatal foreskin. To induce fibrosis, we use high glucose plus TGF-beta or CTGF. To investigate its effects on vascular function, we used female and male rat pudendal arteries. For confocal microscopy, we used HUF cells, we pre-incubated cells with inhibitors for 2 hours, and then to promote profibrotic state, we exposed them to high glucose plus TGF-beta or CTGF for 48 hours. And finally, we added biotinphentylamine or BPA as a TG2 substrate, and we washed away excess, and we detected BPA incorporation with streptavidin alexoflor 488 as a readout of transaminase activity. We also stained for TG2 protein with alexoflor 594, and for nuclei we used TAPI. Here, red signal shows TG2 protein and blue marks nuclei. When we merge the channels, you can see TG2 is expressed or present under resting conditions and also in all these treatments. If you look at TG2 localization under resting conditions, it is mainly distributed in the cytoplasm with a little signal in the nucleus. Under profibrotic conditions with high glucose plus TGF-beta or CTGF, TG2 staining becomes much more prominent in the nucleus, suggesting a possible involvement in transcriptional regulation. Here, in this slide, we are looking at TG2 transaminase activity. Again, red signal shows TG2 protein, and green signal shows BPA incorporation or transaminase activity. And merge image highlights where TG2 protein and activity overlaps. You can see under basal condition, red signal is very low. Under profibrotic conditions, it increases and colocalizes with the protein. To measure transaminase activity quantitatively, we used HUF and HDFN cells, and we used the same protocol, but here we quantified BPA incorporation with fluorometric readout. For qPCR, again, we used the same incubation protocol. We measured mRNA level of TG2, alpha SMA, fibronectin, and collagen 1, and calculated fold changes by using 18S as a housekeeping gene. In HUF cells, we observed very small, non-significant changes in TG2 mRNA expression under high glucose plus TGF-beta. High glucose plus TGF-beta or CTGF mainly increased transaminase activity. LDN and ZTON decreased mRNA expression and also transaminase activity. ZTON decreased basal transaminase activity. In HDFN cells, high glucose plus TGF-beta increased TG2 mRNA expression and also transaminase activity. With high glucose plus CTGF, we did not observe any changes. Then we look at mRNA level of fibrotic markers under high glucose plus TGF-beta, fibronectin, collagen 1, and alpha SMA were increased in both cell types. ZTON decreased these changes. LDN showed a similar downward trend in some of these markers and also decreased alpha SMA level in HUF cells. For ex vivo experiments, we isolated pudendal artery from female and male rats and homogenized some segments and used them for Western blot to assess TG2 protein expression. We mounted other segments in wire myograph to investigate vascular reactivity to LDN or ZTON. For relaxation responses, with or without incubation, we first pre-contracted arteries with phenylephrine and then applied vasorelaxant cumulatively. We express relaxation as a percentage of contraction. For contractions, we applied contractile agent cumulatively, and we expressed contraction as a percentage of high potassium contraction. We observed